So we've built these chromosomes, we've replicated them. Now the thing that we've got to figure out to do is how to distribute them. So we're going to segregate or distribute the DNA. And we need several different things to happen for this. So first, we need a physical capture. So we're going to actually distribute the chromosomes. We need a physical attachment to capture the chromosomes and to capture each chromosome. So we've made them, but if I just spill them out of the nucleus and they're floating around the cytosol now, they're not going to go anywhere. They're just going to sort of float around in different ways. So we need to reach out and be able to just grab onto them and hold them. Okay. Again, we've replicated these. We've held them together. But we need to make sure that these two chromatids, these two things that we've built on, are not holding over here. I'm going to hold them both this way or pull them both that way, or to just have one attachment and that's it. I really need to be able to grab onto both of these and pull them in opposite directions. Okay? And so for this, we need to ensure that these paired sister chromatids are attached to opposite poles. So the poles are the two ends of that spindle apparatus structure made of microtubules, and one chromatid has to be associated with one side. The other chromatid definitely has to be associated with the other side, and it's this organization that's really central. Okay. In a cell, we're going to watch a movie of DNA moving around in a second, and it's not just enough to capture them, you have to organize them. Okay? And so you actually need, in most cells, to bring the chromosomes to the middle. To align them in the middle. And ultimately, we're going to want to bring them to the sides in even ways. And so as a starting line, bringing everything to the middle of the cell is a great way to be able to achieve this. And so this middle of the cell is going to be called the metaphase plate. And then finally, we need to actually distribute them, which is that we need to separate the sisters, as we've talked about through the action of that enzyme called separase. So we're going to take these sister chromatids, we're going to separate them, and segregate away from each other to the new cells, to the new sides that are going to form the new cells. Okay. So classically, there's actually a terminology for these different phases, and I think these names are less important than really these ideas and what they convey. So this beginning part, so if these are all phases of mitosis, these phases here are called pro-metaphase. They're the thing that happens before metaphase. This phase here, where you've aligned them in the middle, is metaphase. It's the middle of mitosis. This phase down here, where you're actually distributing them, is called anaphase. And there's a phase up here that we didn't really talk about explicitly, before we're going to actually begin. It's pro before anything else happens. And this is basically the condensation of the chromosomes and nuclear envelope breakdown. So in most metazoan cells, like our own cells, for example, human cells, the nuclear envelope exists during interphase. It's a pretty solid barrier. You have to have nucleoside plasmic transport, as you've already learned in the class, but it disappears when you enter mitosis. That's not true in all organisms. So for example, fungi, the nuclear envelope never breaks down, and so you still have to build this spindle internal to that nucleus. But so prophase here, you're having condensation. Prometaphase, these initial capture and alignment of events, and then metaphase, they're in the middle, and anaphase, they just go and move apart. So if you imagine that process of moving apart, I've captured them now in metaphase, I've brought it to the middle, I'm halfway between things, and now I want to distribute. How am I actually going to distribute to the two new cells? Well, I've got to move them apart from each other, so I just moved them apart from each other. And if you imagine and just sort of watch my hands and the motions that are existing there, you've got the two poles. One of the ways I could do this is just move my elbows apart. Okay? And that process there is going to be making the spindle longer, and that spindle elongation, and that absolutely happens. And the other thing that you could imagine is that just my arms get shorter. I can sort of mimic it like this, right? In that case, my elbows did not move, and yet the chromosomes appeared to separate from each other. And so we can actually do that with two different physical events. One of them is going to be called anaphase A. And so different cells have different balances of these events, but anaphase A, that is going to be chromosomes moving towards the poles. These are not temporally separated events, but they're different movement types. It's not like one happens before others, but here you can physically move the chromosomes to the pole. And in anaphase B, again, not distinct necessarily, the spindle poles separate. And if you have them move apart further, this is going to help segregate the DNA to the two new cells.